Hi, this is Nick Cap, and I will be lecturing today on basically general microbiology. Let me get a pointer in here. I got this set up all in here. Welcome to Skyline College, and I know you saw me in our first lecture, our first kind of get together. This is going to be our lectures where I will go over roughly a chapter and lecture on it. This is the first half of the second week of our class, so module number 2. I just want to remind you again that this is general microbiology, Biology 240, and a number of you guys are taking the class for different reasons. My name is Nick Cap. How do you get a hold of me? I have an office phone number, but again, I'm not in my office anymore because of COVID. You could call it and it will roll over to my email. It's 650-738-4415. Again, it's always best to call me with email, cap@smccd.net. When we are on campus, I do have an office that's 8226, which is right down the way from the microbe laboratory. My office hours are roughly going to be an hour before lab, 8 to 9, the half hour before lecture, the half hour after lecture. Typically, it's best to kind of email me, and then I can kind of get a hold of you, and we can talk. Emails are best because, again, they leave a written description of kind of what's going on. Normally, we'll have synchronous meetings from 11:10 to 12:35 on Mondays and Wednesdays. That's an hour and a half, 3 hours a week that you'll be with me or see me. We'll have the course that is on Canvas, and you can take a look at it on Canvas. And again, I will be trying to look at attendance and promptness. I didn't necessarily want this to be a distance education course. I really want to have a lot of kind of interaction with you guys. I like students. I like seeing you. I like seeing kind of what you do. And while I do tape my lectures, I really want to have this kind of interaction in class. And we're doing what's called flipping the classroom. And so, you kind of gain the knowledge beforehand, and then when we come together, we try and use that knowledge. And hopefully it's going to help you understand this class a little bit more. And I think it's very important that you know and understand this class. For me, it's 10:26. I have up to an hour and a half to lecture on this, but I want to go as quickly as I can. Materials that you require for this class, again, this slide deck is available on Canvas, and you can take a look at it. Our textbook is online, and it's free for you. You want a lab coat that you can bring in, but when you're doing the labs at home, I do want something to separate you from your lab materials. You also need some lab safety glasses as well. I just happen to not be wearing mine. Again, you need a safe spot in your home to do your lab, and we're going to survey that a little soon, and we will take a look at this. As you can see, this semester is not like any other semesters. How can we remotely have in a class, kind of this college experience? To me, the college experience was going to a classroom, seeing a professor, having people sitting right next to you. Again, this semester we don't have that, and we're going to have something like that, but it may actually change. What I ask of you to be to be patient, and again, overall, try and participate. If you go in class, you participate. You give subtle hints on your head and that. You may gasp when somebody says something. So there's always that kind of interaction. I want to see if we can get that interaction in our classes here. Evaluations are what I call assays. I'm going to try many different ways to see how you know the materials. And I always do this whether we're face-to-face or in this class. I just don't want to give you exams and quizzes. Again, a distance ed class would be something like where you read a textbook and I give you a quiz and then I can give you a grade from that. I don't necessarily want to do that. Okay, that'd be really easy to do, but again, what I found is that most students don't succeed. There's something else that you need, this kind of human touch, human interaction that you can go. Again, if you can read a text, take a quiz, and pass a class, then I would recommend go find somebody who does that because it's much easier to do. But not all of us can do this. I know I can't do that on everything. Again, what I need to know from you is how best do you express yourself. And so not only am I going to be having exams and quizzes, but I'm going to be asking you to write stuff, to make pictures, and to do other things that show me that you know the materials. Again, we've talked about this before. Is extra credit possible in class? Yes. Science in Action will start in 3 weeks, where you will come and look at a talk. And there's other ways that you can have extra credit, and I will post those fairly often. Participation. I would like you to join the American Society for Microbiology, ASM, the Peritoneal Drug Association, PDA, the NCSS, the National Cancer Society. There's a whole bunch of different societies that you can look at, professional society. I would say pick one and try it. I would like you to answer a question or to ask a question. Kind of shows me what's going on, and I will be there will be different ways that I can kind of assay that and measure that. Again, I will enter the microbe of the month we can have. The attendance and meeting lecture is an handwritten report. Again, we're also going to have some field trips that we can do, and that may be interesting. Again, well, some of the lecture materials will change. Basically what this is saying is that I'm going to be giving you 3 midterm exams in this classroom and a final exam. Those midterms are scheduled, and we are going to test you, or I am going to test you on the material that you know at that point. If we don't get through the materials, then what I will test you on is on the materials that we've gotten through. And so I'm saying that the exam dates will not change, although some of the lecture material will change. Student learning outcomes, we talked about this in class, is you want to know microorganisms. Our objectives, what can do is, again, I want you to know the microbiome, etc. And so when we ended our kind of get-together on Wednesday, the question that came up is, what is a microbe? My thesis advisor for my PhD, Marion Hewlett, used to call them bugs, and I would get so mad at her at this. It's like, they're not bugs, they're different things. Bugs are microbes. Bugs can be microbes, but not all microbes are bugs. But, I'm being kind of pedantic there. But anyways, really a definition for what a microbe would be are things that are smaller than 0.1 millimeter. And so 0.1 millimeter, a 0.1 millimeter would be a ruler. I just don't happen to have a ruler here. Those are really small marks on the ruler, and 1/10 of that. And that's basically the resolution of the human eye. And if you can see, and basically anything that's below that is almost invisible to the naked eye, and you can't see it. Again, microbes include bugs, things, germs, viruses, protozoans, bacteria, animalcules, and small suckers. Anything that you basically can't see with the naked eye, but it's there. To know something, to name something is kind of to know something. And again, in biology, the first things that we really kind of started naming in a very specific way. Now, science is there's a cluster of people who are scientists who are all over, and they are different races, different creeds, etc. Hopefully we're better. We're not. We're human beings, and we have all the failings of human beings. But a guy named by the name of Carl Linnaeus, back in 1735, got this idea of kind of naming all living things. He figured he can be closer to God. Everything that he named would be closer to God. And again, how he named stuff is by looking at things that are similar. And again, if you were shaped like this, you were similar, and he would give you a similar name. So we have common names for things, and then we have what's called the scientific name that everyone knows. And typically this is what we call a genus species. By custom, once mentioned and abbreviated, an initial of a genus followed by a specific epithet can be given. And so Escherichia coli is the name of a bacterium that we would look at, but we tend to call it E. coli. That was one of my favorite organisms when I was an undergraduate. Bacillus subtilis, which I worked on as an undergraduate, we tend to call B. subtilis, etc. So we call these things kind of different names. But again, in a paper, you want to give it its full name at least once so that we all know kind of what we're talking about. When two organisms share a common genus, this may indicate relatedness. And so what I mean by relatedness is not only do they look the same, but their genes may be the same, their biochemistry may be the same, the action may be the same. So if one of these genus is susceptible to an antibiotic, another one may be. And so that's really powerful, because what that would allow us to do is once we identify an organism and it's related to other organisms, we know a whole bunch of stuff, probably, about that organism because we know what it is. Again, you look at different species, different strains or races of dogs. And so if you have a Weimaraner, which my brother had, or if you have a purebred hunting dog or tick dog or whatever, they are going to share some characteristics. I didn't do the best at some of this. But again, when two organisms share a common genus, we indicate their relatedness. And again, Carl Linnaeus and all biologists recognized this back in the 1700s. Why do we study microbiology? In the first part of your book, we look at it. So microbes are related to all of life. They are the simplest forms of life. They are in all environments. And I like to say that microorganisms are in the highest points of the atmosphere that we've looked down to the deepest holes that we've ever dug. Do all holes have microorganisms? No, but when we've looked, they've kind of been there. Many microorganisms have beneficial aspects. In fact, the vast majority of microorganisms really don't kind of see human beings. We're kind of totally different from them, and they don't cause disease. While we may focus on disease microorganisms, don't touch that. That has germs on it, but those germs a lot of times have some characteristics that may help us. So microorganisms have many beneficial aspects. In fact, a lot of the air that we breathe actually comes from microorganisms, algae that are growing out in the ocean. If there wasn't that algae in the ocean, and then even though the trees around us, and there's lots of, I'm in a forest in here in Pacifica, and even though there's lots of trees, we think of the tropical rainforest producing lots of oxygen. Actually, more oxygen is produced in the ocean by microorganisms. Again, microorganisms have a relatedness to life process, and that is food web. A lot of what we eat depends on microorganisms and nutrient cycling. And so again, we'll talk about these things a little bit later. But the carbon, phosphorus, and nitrogen that you have is cycled in the process. Again, microorganisms are related to our life process. So when we study the biochemistry of microorganisms, we're studying our own biochemistry. And again, we can chop up and divide microorganisms many times, and people don't care. We chop up a human being, you got to go to jail. I don't know, that's hard. But anyways, only a minority of microorganisms are what we call pathogenic, that they cause disease. This right here is kind of showing you here one of the things that kills you all pretty high, besides accidents and gunfire and stuff like that, is something that we call meningitis, and that's an infection of some of the tissues inside of our body in the nervous system. And one of the things that we can do is that when you have meningitis, and that is kind of slurred, somebody who has an onset very quickly of kind of slurred speech and a number of different things like this, difference in behavior may be an indication of meningitis, or they may be drunk or something like that. But meningitis would be the disease. There's different ways that we have of treating this. Viral meningitis is different than bacterial meningitis, and how we know in this particular right here, this is called a spinal spinal tap. They can go in with a needle, and you can take some of this cerebrospinal fluid, and you can look for microorganisms in there. And I would say here that this is clear, and this may indicate either a normal material or viral. We tend to see it when it's cloudy. It tends to be a little bit more, and that works on this a little bit more. And so this is one of the ways that we diagnose, and that's how we see a microorganism is present. Another thing that we tend to look at when we look at microorganisms are what we call emerging infectious diseases. And we have one going on right now. I mean, this COVID-19, that's the name of the disease that's causing, wasn't here a year ago. We didn't even know about it. And so it's this thing that kind of came up, and we'll talk about that a little bit. These emerging infectious diseases are very important because we can actually use them. Imagine, we're pretty sure that this one here was a mistake, but imagine if we wanted to do something to kill out. And one of the reasons why we went into Iraq, when I was younger, 20 years ago, was because we thought Saddam Hussein, who was the leader then, may have had biological weapons. Again, emerging infectious diseases can become due to new evolutionary features. This idea of kind of what is evolution. Evolution is something that's going on, and we'll talk about that a little bit later, but microorganisms are continuously changing. And so what we're seeing is that microorganisms are adapting to their environment. Again, I don't know if you know this, but many of our antibiotics aren't as useful as they used to be, and this is kind of because microorganisms are evolving and gaining these particular materials. And again, emerging infectious diseases may be a response to encroaches on the environment. And so we are going and living and communing with different areas in the environment that we kind of never did before. And so that means human beings and these microorganisms are interacting with one another. We would like those interactions to be positive, but they aren't always positive. Again, we're going to do this in class, but can you give me an example of an emerging infectious disease? We'll have a discussion on that, so we'll talk about that during class. Microbes are used in research. Your body contains 10 trillion human cells, and you have 100 trillion microbes. And so you have your body is basically a symbiosis of microorganisms and human cells. Microbes are really good to use in research because they're easy to grow. The biochemistry is essentially the same. A lot of the biochemistry, DNA replication was all figured out in microorganisms first, and they are relatively simple and easy to study. This topic that's near and dear to me, which is biotechnology, is the use of biological systems to produce useful items. The use of biological information to make things or improve the human condition. And so biotechnology has really kind of helped this pandemic. Imagine this pandemic happening 300 years ago. It would be kind of the wrath of God, and we wouldn't know what it is. Imagine this pandemic happening actually 30 years ago. We would have this bacterium, and we have no idea of what it is. But because of biotechnology, we were actually, once this disease was first identified, we had the sequence of it in like weeks. Okay, and we're going to have an antibiotic. Actually, we're going to have a vaccine for it, hopefully, in a few months. And actually, we have vaccines for it already, but we're in the process of testing them and seeing if they work. And that's one of the things that we could talk about a little bit later. But basically, when this biotechnology, our knowledge of biotechnology, our knowledge of these microorganisms builds on one another, that we're going to be able to do things a lot faster than we ever did before, as well as use microorganisms to make things that we need. In this slide here, we're talking about the diversity of microorganisms. Microorganisms are very different. So what we call microbes consist of a number of different things. And so bacteria are what we call the single-celled prokaryotes. Protozoa are eukaryotic, single-celled, clonal, and have many forms of nutrition. Fungi have an absorbent lifestyle. They're single-celled and filamentous. Viruses are acellular entities. We can talk whether they're alive or not. Other microorganisms include worms and insects that are small, or they have parts of their life cycle where they are microscopic, less than 0.1 millimeter across, that you can't see them. When we looked at life, Carl Linnaeus, when he looked at life, he basically said, there's animals and plants. And he actually looked at rocks and minerals and stuff too at the time. That's what a lot of naturalists did. Ernest Haeckel actually divided up into Monera, which are basically the bacteria, the protists, the eukaryotic organisms, plants and animals. And then Carl Wohl's in 1969 separated out into Monera, fungi, animals, and plants. And actually, we severed that. That's called the Five Kingdom century. We've actually divided it up a little bit further than that to say that, it's the... we've actually divided a little farther into that, kind of looking at domains, and we'll talk about that a little bit later. Remember, this is an intro. I'm showing you this stuff, and we'll show it a little bit later. These are basically the three. These are the three domains of life. And they're all different. And again, how this came about is we got new technology, and we're able to look at life in different ways. And again, when Linnaeus looked at life, he could just kind of see stuff. Oh, that's a plant, that's an animal, that's a plant because it's green and it doesn't move, that's an animal because it moves and it's not green. We put up more information. And so when we got this more information, and we looked at relatedness, we actually found out that in life we have the eukarya, which would include trees and us and fungi and slime molds and amoebas. There's archaea, which are the ancient bacteria that grow in these areas that we kind of don't know. And the bacteria, we'll talk about their relatedness a little bit more. But again, by looking at proteins, looking at DNA, we can see that life was divided, and that once where we used to think about the big difference between plants and animals, we realize that as living things, we are a lot more closely related to a plant than we are to some of these bacteria. And so we'll look at it. So wolves and foxes developed this phylogenetic tree, and they did that at the U of I, which is where I'm from. Naming of microorganisms. To name something is to understand it. Possibly, I think we'll look at a little bit. Do I have something named after me? No, I have a friend of mine who's discovering insects and stuff like that, and so he's named stuff after people. He hasn't named one after me yet. I think I'm going to kind of get on him on that. A lot of the microorganisms that we have are named after people who discovered it, are named after kind of the bosses or somebody you can honor by doing that, or they're named from the area, and we can give examples of that. The old-time piece of equipment that would allow us to identify stuff is something that we call the Bergey's Manual. And this would allow us to give the name. That's the old standard for identifying microorganisms. We have a problem with that. I have a problem with that. Do you know that most of the microorganisms that are out there, we can't grow? We can't characterize by the usual ways. So in Bergey's Manual, what you do in the Bergey's Manual, you'd look at the biochemistry, or you'd look at it underneath the microscope, and you'd say, oh, this is a gram-positive microorganism, this is a gram-negative microorganisms. You can separate. Most microorganisms we can't identify that way. And so the Bergey's Manual is kind of going on the wayside, and I'm going to talk a little bit about that and what's going on with that a little bit later. But I do want you to know this thing that's called the Bergey's Manual, which is sad. In the lab, we have a whole bunch of Bergey's Manuals, and they're actually not printing them anymore, so to find a hard copy of it is pretty good. So let's take a look at these microorganisms here. This is figure 1.2 in your textbook. Again, 0.1 nanometer is roughly an atom. We'll talk about these sizes a little bit, but basically between basically 10 nanometers and 1 nanometer we'll have these things that we call viruses. Bacteria are between 100 nanometers and 10 micrometers, and then eukaryotic organisms that are in here. The human egg is just slightly less than 0.1 millimeters. So you guys have probably never seen it, but if you're able to look real close, you should be able to see a human egg with the naked eye. We'll talk about these a little bit, and we'll talk about this table a little bit more. I've been talking now for about 20 minutes, and probably about 20 more minutes worth of talking to do. When we look at microorganisms, a lot of times we talk about the morphology. That is kind of what they look like. And, again, human morphology is basically looking at an anatomy. And so when we look at microorganisms, first of all, we looked at microorganisms underneath the microscope, and there's really not a lot of difference. You have, again, you can tell a difference between a tree. You can tell a difference between me and my wife. You can tell a difference between a dog and a cat because of these characteristics, especially the morphology of their shape. For microorganisms, you don't have that, so you're going to need to know some of these names. A cocci is a microorganism that is round. A bacilli is a microorganism that is longer than it's round, so it's shaped like a rod. A vibrio is a rod-shaped organism that has a little bend to it. A coccobacillus is a smaller rod that's almost a coccus, but it's bacilli. A spirillum has kind of a twist to it. A spirochete has a definite helical in there. And so I will want you to know what some of these morphologies are, and ideally we can do an activity on that as well. In this slide, I'm going to keep going. I need to keep going because I don't want to take all this time. But this is basically a slide. This is not from your book. This is another picture that I had showing the different types of microorganisms that are out there. And so again, we have bacteria, we have fungi, we have algae, we have protozoans, we have viruses, etc. here. Sorry, I guess I should have put the COVID-19 virus. I'll try and put that in there. I'm going to real quickly go through the different types of microorganisms. We're going to go over this again in a little bit more detail. Again, this is the introductory, and I was just going to show these up. Bacteria include prokaryotes. They have the cell wall that can consist of what we call peptidoglycan. They divide by binary fusion. Basically, they get big, they split in half in those two microorganisms. For energy, they use organic chemicals, inorganic chemicals, and a number of other things like photosynthesis. The archaea literally means the old ones. These are prokaryotic as well. They lack peptidoglycan. They tend to live in extreme environments. And these include the methanogens, the extreme halophiles. So again, if you're driving across the Dumbarton Bridge down there and you used to see these ponds that were bright red, that was the salt ponds that were in here, the archaea growing there. The extreme thermophiles. These are microorganisms that can grow in boiling water. Okay, kind of scary. The next set of organisms that we'll look at is the eukaryotes. We'll cover what eukaryotes are a little bit more. The simplest eukaryote is the fungi. Their cell wall contains a molecule called chitin. We'll go over that a little bit later. That is a polysaccharide. They use organic chemicals for energy, so they break down things. They are not photosynthetic. Molds and mushrooms are multicellular, consist of masses of what we call mycelia, which are composed of the filaments that are called hyphae. These are some words that you need to know. Yeast are unicellular fungi, and so fungi are important. Let's go over this. This is a bunch of oranges here, and this is the molds that you can kind of see. Fungi are really interesting organisms because they compete with us for food, and they can be pathogens on human beings. They're really good at it, and you know what? We don't always win. One of the scariest things, I think, a scary infection that you can get is a fungal infection. This here is actually a pool from Yellowstone National Park called the Morning Glory Pool, and this is teeming with bacteria. And yet that water is almost boiling. You can kind of see the steam come off of this. These are kind of things, if you were to jump in there, you'd be boiled just like a lobster, and you wouldn't survive if they didn't get you out of there right away. I've heard stories about people like, oh, it's a hot pool, let's jump in it, and they all die. Some life can live in spots that might exist on other planets. It's really kind of interesting. So these are characteristics that we're going to look at. The life that lives in here are totally different from us. It's not the same, and we're totally different from them. The great thing about this is those organisms aren't going to be pathogens, and also the great thing about it is these organisms may have some characteristics that we can use for our benefit in biotechnology. Protozoa are eukaryotes. They absorb or ingest organic materials. They're very simple forms of kind of animals. They may be mobile via pseudopods, cilia, or flagella, and we'll talk about those characteristics a little bit later. Algae. This is one that hopefully we'll get a chance to look at. This is called the Volvox. It's one of the most beautiful organisms underneath the microscope that you can see. Algae have cellulose for their cell walls as well as other carbohydrates. They use photosynthesis for energy. They produce molecular oxygen and various organic compounds like sugar. If it wasn't for the algae, like I said before, most of the oxygen that we breathe actually comes from algae, and a lot of the food stuff that we eat, the fish in the sea, all depend on algae to get their carbon and chemicals. Viruses are what we call acellular parasites. They consist of a DNA or an RNA code. They can't live on their own. Given raw materials, they don't have it. They don't necessarily have a metabolism, yet they use you or I. And so this is looking at the HIV virus. Right now we're really concerned with the SARS virus. It's a core of a nucleotide, either DNA or an RNA. SARS is an RNA core that's surrounded by a protein coat. The coat may be enclosed in a lipid envelope. The viruses are replicated only when they're inside the cells. And so when you sanitize an area and you put some alcohol or something on there, basically what you're doing is you're destroying the coat for the COVID, and it's no longer infectious. It's kind of one of these weird things. If you were to look for the RNA, you'd still see the RNA, but it's no longer infectious. We as scientists are constantly monitoring the environment for these viruses, and that's one of the crazy thing about I'll try not to get into the politics and science, but it's happening. Politics actually came in and messed with science first, and so we're going to mess back. But basically, every year you guys should get a flu vaccine, and actually hopefully you guys are getting a flu vaccine now, because it can be a bad flu. Basically what we do is we go out and do field work as biologists, virologists. We go out there and we look at what viruses are out there. And what would have been really nice is if we could have found out that this virus is in a place, and stopped it from coming here rather than having it happen. And that's really a case. So some of these viruses actually come from places like China, Iran, and Iraq and South America. We used to have scientists all over looking for these, and now we've kind of stopped kind of funding them, which is kind of ridiculous. And I can talk about that a little bit later. Some microorganisms, just a history of some of the microorganisms that you need to know about it. Giardia, if you've ever gone hiking, so this is the microorganisms over there. It's a protozoan parasite, infects humans and other mammals. You tend to get this when you go hiking and you don't kind of sterilize your food. I've had Giardia, very bad farts, very bad diarrhea. When you're hiking, it's just the most horrible thing that you can get. There are other multicellular parasites that we tend to not see. These are eukaryotes, the parasitic flatworms and roundworms and hematoliths. They have microscopic stages in their cycle. That's why we call those microorganisms, although, again, you're going to be able to see the larger organism, the larger parasite, depending on kind of where it is at in the body. In the United States, typically in first world countries where we treat our sewage, where we don't eat material that's been contaminated with sewage, not all the time, we tend to not see these parasites. But in third world countries, you do a little bit. Here's an example of some of these. The beef tapeworm, we used to have this a little bit. Now we monitor our beef stocks, and so we no longer have this tapeworm, but there's other organisms that live like this. The guinea worm, which is basically to get rid of this guy here, people would actually take the worm and wrap it around matchsticks. It would actually come out in the morning, and you wrap it around, and you would slowly kind of pull this guinea worm out of your skin. That must just be horrible to do. We'll talk about those a little bit later. For all of this, how do we know all of this? And your book doesn't go into this, but I really want to take a few minutes and describe kind of what's going on here. Is that we have something that's called the scientific method. That is where you make an observation, you make a hypothesis, you test the hypothesis, you draw your conclusions, and you repeat. So these are the steps to the scientific method. And right now, the scientific method is hopefully going to serve us. So if, what we have is we have a microorganism that is causing the disease. Our hypothesis is we could train our immune system to actually recognize the COVID, the SARS-2 virus, and so that way we won't get the disease. And so what we're trying to do is we're trying to make versions of COVID or partials or purify COVID and inactivate it so it won't cause disease. So these non-disease forming cases, we're going to take those kind of pseudo-diseases, inject them back into the human body, and see if they work as a vaccine. And then we'll produce a whole bunch of those. And so basically, how we know stuff is we do stuff by the scientific method. Again, doing something once. I said this morning, I hope the sun comes up, and I made the sun come up. That's not the scientific method. Where you do the scientific method is where you take a question. Again, example of the scientific method, and this is something that we'll kind of go over in class, is that you can do something like, well, you may do this every morning. You go out to your car and it doesn't start. What's your hypothesis? Oh, well, I'm out of gas. I could be out of gas. The battery could be dead. I could be using the wrong key, etc. And then you, again, if you're out of gas, what you can do is make an observation and say, hey, I'm going to look at the fuel gauge and see if it's in there. Is the battery dead? Do the lights turn on when I do this? And so there's a number of different things that you do. And so how we know what's going on is we use this scientific method. And again, I want you to use the scientific method to study. And so what you're going to do is you're going to read the chapter and you're going to say, how well do I know this chapter? Well, you're going to take a quiz. And if you do well on the quiz, that means that you know the chapter pretty well. If you do bad on the quiz, then you need to go back and you need to restudy. So that's what we call a scientific method. Some of the requirements that we need for the scientific methods is you need to kind of look at one variable. You can't have a whole bunch of different things. Also, you need to believe that you can solve the problem using the scientific method. If you feel that you can't solve it, then, when things are unsolvable, it's because you think they're unsolvable. You need to have experimental controls, And then again, we can look at this, how to discover things. So we're actually, this is one of the things that we're going to do in class. Is I'm going to have you kind of ask some questions and have some discussions on doing the scientific method. And so again, when HIV first came out, we didn't know if it caused AIDS. And again, how do we know that this particular organism that we're looking at, the COVID-2, how do we know that that causes SARS-2? How does SARS-2 cause COVID-19? Anyways, we'll discuss that during class. And again, this info is not necessarily in the chapters, but I think it's very important, and we will talk about it. Again, our knowledge of microorganisms allows us human beings to prevent food spoilage, to prevent disease occurrence. It led to the aseptic technique to prevent contamination in medicine and in microbiology laboratories. And again, when we look at this, one of our best tools for preventing disease is actually using the scientific method. This has led us to have some things called universal precautions, which are set up by the CDC, which is basically to use gloves, gowns, masks, and goggles to minimize risk of needle sticks, to disinfect your area, to preventative treatment after exposure, to reduce your risk, to treat all patients the same. And again, that's kind of one of those things. And where this kind of came about is, we used to touch and interact with people kind of all the time, and due to HIV, we had basically some patients like, oh well, they might have HIV, let's treat them like a pariah. But really, you should treat all patients the same and treat all patients like they have a deadly disease. And what I said here, hepatitis B is actually, you have a greater risk of dying from that, from HIV, and actually COVID and all these things. And so there's not only COVID, but there's lots of other diseases that are out there. We should practice universal precautions on kind of everyone. We've been kind of sloppy not having people not masked. This problem where people have problems wearing masks all day, you need to work and make better masks that we can wear all day. I'll get off of that. So again, this is the end of my first lecture that I have. There's some slides that are behind this, but I'm going to use that for another lecture. So it looks like here I've lectured for about 34 minutes and done that. And so that's about what I want to do. I don't want to kind of get too much, and ideally I'll get these lectures down to about 10 minutes. And so that's most of the first chapter. So it's everything in the first chapter except for basically the history of microbiology. And I want to look at the history of the microbiology a little bit more. Okay, so thank you very much for attending this first lecture. I now think you are ready to take a quiz. There's going to be some hard questions in the quiz. You can go ahead and look at it. Now that you know the materials, you should be able to do pretty well on the quizzes. And so thank you very much.